Hello, everyone, and welcome to today's webinar, Strategies for HCP-MS Analysis, Designing the Best Approach for Your Process and Product. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SciEx. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Christina Morris, Senior Scientist at BioPharmaSpec. In addition to a question and answer session following Dr. Morris's presentation, today's webinar has an additional interactive component, poll questions. We invite you, the audience, to share your opinion on two multiple choice questions today. The first I will ask in a moment, and a second at the conclusion of Dr. Morris's talk. Let's start with our first poll question. Please take a look at the presentation window to view the poll question and click on the answer you would like to submit. Is MS analysis of HCPs a part of your product and process development workflow? A, yes, we are using a combination of MS analysis and other orthogonal method for HCP identification, such as ELISA. B, no, we will only use a generic ELISA kit for HCP identification. C, no, we will only use custom-developed ELISAs and/or ELISA kits for HCP identification. Or D, we are not currently using MS analysis for HCP, but we are considering implementing this as an orthogonal technique. Give you a few seconds to make your selection. And it looks like D, we are not currently using MS analysis for HCP analysis, received the most answers. We appreciate your participation. Dr. Morris, you may now begin your presentation.
So hi, and thanks for the kind introduction. Good morning and good afternoon to everyone. So our talk today is on strategies of host cell protein analysis using mass spectrometry, where we're specifically focusing on ways to design the best approach for your process and your product. There's just a little bit of info on us, BioPharmaSpec, as a company. We offer and specialize in biopharmaceutical characterization services, primarily with mass spectrometry, but also using other instrumentation. So we've chosen to help fulfill the different regulatory guidelines with these different instruments, and specifically for biopharmaceutical products. As you can see on the slide, these include biosimilars, ADCs, mAbs, AAV-based therapies, and of course, product impurities. So our staff have in-depth knowledge of the instrumentation, and some of them have over 35 years of experience in some cases. And we're directly involved actually in pioneering the approaches for MS biopharmaceutical characterization. We also have a strong research background, and some of the team, in fact, were involved in a recent SARS-CoV-2 publication on the characterization of glycosylation of the spike glycoprotein. So this experience gives us the right skills for troubleshooting and method development, and that's proven to be useful for clients in overcoming any product-specific hurdles. So currently we've got two labs, one in the UK and one in the US and Pennsylvania, and we've got agents in the UK, in India, Canada, and Korea. So our labs have dual capacity in the sense that there's duplication of instruments and methods, and that allows us to give our clients greater flexibility in case of instrument breakdown or overflow. And it also allows us to stick well within client schedules and to meet tight turnarounds. So if you have any questions about our services, please don't hesitate to get in touch. My details and my colleagues' details will be provided at the end of the talk. So why are we monitoring for host cell proteins? Most of us, I think, are aware now of the need to monitor the host cell protein population in a biopharmaceutical drug substance. It involves multi-step purification procedures that polish most but not all impurities out of the final product. And manufacturers need to determine which and how many HCPs are present, and to know that the safety limits are product specific and defined on an individual basis. During manufacturing, the different stages of validation continually cycle between identification of process variables or critical parameters, defining control strategies, monitoring, and then making appropriate improvements where necessary. And what we know is that process-related impurities like HCPs are highly dependent on the process and the controls that are applied. So for any given product and process, it's important to continue to monitor the stages for any changes that can impact the HCPs detected, and to do it in a timely manner, because of course what's suitable for one product may not be suitable for others, even using the same manufacturing platform. And then it's also recommended that you monitor HCP numbers throughout the different purification stages to show that there's clearance of impurities, and in doing so better understand the control strategies used. And, you know, as a consequence of that, encourage improvements that minimize levels of HCPs. So targeted monitoring of HCPs are mainly focused on ELISA-based methods, which are the gold standard. And more recently, thanks to advances in speed, sensitivity, there are now also MS-based methods available. We run these alongside the HCP ELISA tests, as they provide an increased breadth of HCP identification. They're highly reproducible and specific, and they're proving to be an excellent orthogonal method for the depth it brings to the characterization and of actually understanding the HCP profiles. So given that this is an HCP-MS presentation, I've highlighted a couple of MS-based quantitation strategies, and these can be split into two main groups: those that use labeling technologies and those that are label-free. Labeling methods will include techniques with multiple approaches within them, but they broadly consist of two categories: post-harvesting strategies or metabolically active labeling. So post-harvest tagging strategies introduce labels at the protein or peptide level after the cells have been lysed, whereas the metabolic labeling involves endogenous incorporation of stable isotope labels in active cells. So these labeling techniques can tag peptides metabolically, enzymatically, and chemically, and have been primarily developed for monitoring differential protein expression. And I've included a few examples of the techniques in the blue table below. So MS labeling methods do provide greater accuracy for quantitating proteins of interest, but increasingly it's the label-free quantitative approaches that are being employed to replace or complement the labeling methods, and that's just due to the ease of the experimental setup and the relatively low cost. So the technique also identifies a larger number of proteins with a wider dynamic range of detection, and the workflows, as I said, are simpler, they're faster, and for example, there's no need to determine labeling efficiency. In HCP analysis, we inevitably are aiming to identify and quantitate very low abundance proteins. And for that reason, it's why targeted mass spectrometric techniques have developed. So using both labeling and label-free methods like multiple reaction monitoring or sequential window acquisition of all theoretical mass spectra, so also known as SWOT-MS. So using these methods, we've produced the best results really for these types of analysis, and that's why they're frequently adopted for HCP-MS study. So I've included here a quick summary of a typical HCP-MS workflow. In this case, we're using a SCIEX-based method to show the HCP detection strategy. So we start with the reduction, alkylation, and digestion of all proteins within a mix. Then we'll go on to online LC separation, and then finally the mass spectrometric identification and quantitation using a high resolution instrument like a quadrupole time of flight mass spectrometer. So looking at stage one, we have the library, or we conduct the library creation. So a host cell lysate is commonly analyzed with IDMS methods to build the HCP reference spectral library. Bioinformatics tools use a species-specific database of the host cell that then identify potential protein matches in the IDA data. And then we'll switch to SWOT-MS, which is what you can see in 2A and 2B. So here you've got data-independent acquisition methods that have greater sensitivity, and these are used to screen against the host cell reference library for any of the matching HCP identifications that are in your samples. Standard spikes are used for the relative quantitation. So these are known standards at known concentrations, and they estimate the mole values of the observed HCP areas. So then lastly, for stage 3, if you like, depending on your specific process or development strategy, there is the option to use a higher accuracy quantitative approach. So for example, the synthetic heavy labeled peptides and a triple quadrupolar orbitrap instrument are commonly used. Here the HCP peptide concentration is calculated by comparing intensities of the heavy labeled internal standard, which is the analogue of the HCP of interest, with the native peptide equivalent that's in the sample. So for those that are interested in the MS acquisition, just to show you a little bit more detail on the MS front, regarding the 2 MS run types that are performed, we've got the IDA-MS, which I've already mentioned, which is used to create the spectral library, and then we've got the SWOT-MS, which is used to screen the library with the sample data for quantitation and ID. So the top image shows the information or the data-dependent acquisition, where the method cycles between the survey scans and then subsequent targeted MS fragmentation of a very specific precursor signal that's been identified in the survey. So as the fragmentation spectra are attributable to a single precursor mass, it's used for the library generation. So SWOT-MS instead uses a first quadrupole isolation window to step across the mass range in the MS survey scan. And then it collects MS fragmentation data at each step to generate fragment ions from all the detectable precursor masses. So although the MS assignments are more ambiguous, it's more sensitive, it's reproducible, and it gives us a more complete picture of sample complexity, and it's for this reason it's used for the quantitative aspect of the process. Right, so the focus of this presentation is to highlight some of the options or considerations that should form part of an HCP-MS strategy. And we thought it might be helpful to think about as part of your process development and monitoring. So I've already touched upon there are different ways to monitor proteins of interest with MS, including different quantitation strategies and instruments. Over the years at BPS, we've adopted specific approaches to maximize our efficiency and to optimize our detection capabilities for client projects. And in doing so, we've noted that there are 3 key areas which we think warrant the extra attention. And these include the spectral library, otherwise known as the protein or reference library, then the sample selection process, and finally consideration for data optimization or data quality expectations based on the analysis methods and the data processing that's being conducted. So I will now talk you through them. So first of all, what are our HCP library options? As I've summarized, IDA-MS data collects the information about the HCPs with high confidence. The bioinformatics searches match predicted peptide sequences in your data against sequences in a species-specific database of the host cell, and then assign a confidence score for the match. So this step, as you can imagine, can be quite critical to what HCPs you might identify later on in your sample analysis. And there are a few common options when it comes to selecting the type of library to use, each with its own advantages and disadvantages that I'll go through. So option 1 or type 1 is when a library is made from a host cell lysate using a null cell or a host cell lysate that contains the drug product. And that's used to form the basis of your library and will produce host cell proteins that are present in higher concentrations. So this is especially useful for the data processing and review. IDA-MS provides more confident results, but the sensitivity is not as great as SWOT-MS because the precursor ion selection for fragmentation is usually in the order of the most intense signals. So we benefit from using strongest possible samples. So unlike pre-existing host cell line standards, the other advantage is that the spectral library will be specific to the purification process for your drug. So that's important because as we said, the smallest changes in the manufacturing can affect the HCP population. The downside is that the lysate will need to be generated, and it's not always that straightforward. Specialist equipment and methods may be required, and these might not be normally part of a company's in-house setup. So due to the complexity of a typical lysate, there is also the added cost associated with the library generation, also to ensure good LC separation and detection of low abundance HCPs on our side. A second or another option is the use of in-process samples and or drug substance, final drug product samples for the basis of your library. So you can use samples to build your library, you don't need a lysate. This can be useful in cases where in-depth analysis of the low ppm or low abundance HCPs is not necessary, and an overview is preferred. So, or perhaps like a lysate is difficult to generate. So the advantage here is that the sample preparation stages can include both the library prep and the HCP quantitation, and that can significantly cut the analysis related costs and the project timeline. And lastly, we've listed a third type of spectral library, which can be generated using off-the-shelf host cell standards, so something you can buy. And this provides the advantage of having a greater depth. And in cases where a company has a selection of different pre-existing host cell libraries, it will also reduce the expenditure associated with the setup of the library. But you've got to be aware of the downside, which is that it's not going to be process specific. So I've got a couple of examples in this slide to demonstrate a few points about library types. At BPS, we do like to dedicate project time to generating a library and to ensure that it's as broad as possible, and that it just gives us greater confidence in the low-level HCPs during data processing and review. On the left is an example of the protein yields generated from a protein pilot sequence database search. So IGAMS runs have been queued up for amino acid sequence matching by the software. And they also apply decoy sequence database searches to select a false discovery rate, also called the FDR. So the FDR is indicative of the number of false positive protein identifications that are likely to be found in the database. And the protein yields in the spectral library are reflected by the FDR percentages that are selected. So you can see here that the different yields for the percentages that have been given. So 1% global FDR is a standard statistic, and in certain cases we have up to this to 5% in order to increase our library size, but that comes at a cost of increasing the likelihood of false positives. And then to the right, we've presented protein pilot results from 2 different library searches, just to illustrate some of the parameters that we like to look out for when building a protein library. So in this case, the bar charts both display the number of HCPs that have been observed at different peptide counts. So in order to produce more confident data, we want to see ideally a higher protein total with larger counts, so more proteins with higher peptide counts. And what we can see here is the counts are very high in the top chart, which is from an off-the-shelf lysate library. But as you can see, it's not the same phenomenon. It's not been observed in the data below, which was produced from in-process samples. So for the latter, the library protein and peptide numbers are going to be very dependent on the sample types received and the number of samples as well that you're using. So what we're doing here is we're showing how representing the data in this way and monitoring for this is a useful way to assess the quality of a library and something to bear in mind as a guide during the planning process for your analysis. Following library generation, sample analysis then takes place. And that's where we're screening our sample data for HCP IDs and then quantitating hopefully down to low ppm amounts. So deciding which samples are appropriate for quantitation will be specific to your aims, and we've got 3 strategies here, you'll see, with different sample selection rationale that might assist in this process. So the first one is what we like to call an overview analysis. So you've got 1 or 2 process stages that can be selected. So for these types of strategy, perhaps you're not yet happy with certain stages of your process, you want comparison with an existing ELISA, and to focus on a very specific stage to monitor improvement. So that might involve quantitating multiple batches, for example, or lots, to check for any changes, overlap, clearance, etc. Or alternatively, you have low HCP numbers, perhaps validated with an ELISA, and you're happy with your upstream process, but you just want an orthogonal check of the downstream stages. So here you can focus on just the drug product or drug substance, or anything really of concern actually. And then we have a full assessment scenario. So using samples for multiple purification stages, all the way from upstream in-process samples down to the drug product. So here you might not have a reliable ELISA, and therefore there's a heavier reliance on other available orthogonal methods, or perhaps you may have changed an upstream processing step, risking changes in the HCP population. For example, your custom process specific ELISA needs to be redeveloped. So that will push time constraints. And in these situations, then MS is actually a really good way to ensure, in the meantime, that there are no changes or new issues and that good clearance has been achieved. Or maybe there's also the possibility that you're at the final stages of your product development and showing clearance anyway is an important box to tick. So we like to emphasize this at PPS because we think monitoring HCP levels throughout the process optimizes the understanding of your control strategies. And of course, those can significantly impact timelines further down the line. And then the third option is what we call, again, an accurate assessment. So in these cases, you do need to know what the HCPs are that you're hoping to quantitate. So if there are specific HCPs of concern, or perhaps that are higher than expected quantity, and you would like a more accurate method of assessment, then labeling MS techniques, such as the use of heavy label analogues of HCPs, are a good option. More commonly, this technique tends to be performed on the final drug product, but if there are other stages of interest, they can also be analysed. Another benefit is that you may be unsure about a previously used MS relative quantitation method for a drug product. So these techniques are going to provide more accurate results, and you'll just have better confidence in the numbers that are calculated. So MRM programs like the SciEx one displayed are powerful tools to do this. They have the ability to monitor multiple transitions in a single LC run, and by using short retention time windows, they can maintain the high sensitivity screening. So the final topic of discussion is related to data review and points to bear in mind once you've produced your HCP results. Data processing by proteomic software is required to provide the HCP matches against the spectral library and quantitate data sets in ppm, either micromoles per mole or micrograms per gram. So how much data is generated and the quality of it is largely dictated by how it is processed or interpreted, as well as the LC-MS methods employed. So we've split this topic into two broad sections: the software considerations, or how you are processing your data, and then considerations for the LC-MS strategy also. So if we start with software and automated data processing, we know that the bioinformatics tools are fundamentally important at filtering such enormous data sets, which usually comprise of many replicates. Sometimes you can have thousands of HCP identifications. And the algorithms that sort through these spectra are quite, well, they're critical to the HCP-MS strategy in terms of time efficiency. But as with any method, there are limitations that can lead to HCP-MS misassignment or false positives. And it's important just to be aware of those. So we've listed a few. So for example, underestimating peptide confidence scores, leading to a protein being excluded from a data set altogether. So in these cases, it's often due to a weak library. So it's just something to bear in mind when selecting your method for the library creation. Another is misassignment of HCPs that's due to unexpected proteolytic cleavages normally. It's difficult to predict these sorts of circumstances. They're more commonly seen with drug product peptides, which have been misassigned as HCPs. So if a drug product has peptide modifications or unusual cleavages, it can be falsely picked up as a high intensity HCP. Another one is peak picking errors, especially for low abundance HCPs where the wrong area has been selected within the retention time window, and it can lead to high statistical errors across replicates. So linked to this are retention time alignment issues. So if you're trying to align gradients from, say, a standards reference library with a spectral library from ELISA, you've got to be careful about the alignment process to ensure there's enough information, retention time points to match the gradients, because it can it can then really impact the HCPs picked out according to the retention time search windows that you've set. And then finally, I've got a point there just so that I make a note about our reliance on automated data reviews. So it's simply consideration that there are advantages to carrying out manual reviews in order to supplement the automated hits. So algorithms won't catch everything because of the challenges of interpreting data over, sometimes up to 5 orders of magnitude sensitivity range. So further manual or human scrutiny of any HCPs can have a significant impact on the total estimations at the very end. And we think it's very valuable. So as I said, data quality is also dictated by the LC-MS strategy. And I'll just go through a couple of points. In the next slides, I'll go through some examples and for the moment, list a couple of the factors to consider. So these are to consider, but they're not limited to, for example, how many technical repeats have been used. Yes, reducing repeats may cut down cost and timelines, but more often than not, more data will improve statistical confidence and ultimately provide further evidence for low abundance HCPs. Another is whether you have supplemented the base library with sample IGA to account for any HCPs missing from the library and to avoid further false positive results, for example, or missing something altogether, as I said. So again, there's more time involved in this process, but there will be higher confidence in your data. Then some HCPs might be suppressed altogether. So if you've got a particularly busy lysae or sample type and your 1D LC-MS method has poor separation or is prone to, say, high pressures, etc., you might want to switch to a 2D LC-MS method. It'll be more time consuming, but you're more likely to pick up the low abundance intensity HCPs and generate better data and just have a more complete representative library. And then a final point really to be aware of, as mentioned previously, is the accuracy of relative quantitation. It does have its limitations due to ionization differences between the spike standards that you've selected to help in the quantitation and then the actual HCPs themselves. So the peptide ionization efficiencies and responses will probably be different. And so a more accurate evaluation of the HCP amounts can be performed using an MS strategy, such as with cold heavy labeled isotope analogues of an HCP. And yeah, you can adopt this as a follow-up method. In the following slides, we've presented a few examples of how the LC-MS method can sometimes significantly impact the data quality. In some cases, the effort and cost are not valid, but if a client is more heavily reliant on the HCP-MS or you need to troubleshoot issues in your purification process, then optimizing your methods and monitoring the results will be more worthwhile. In this particular example here, to the right, we have data from an off-the-shelf E. coli standard lysate. So where we show the relationships between the number of IDA-MS runs, the column load and LC gradient versus the number of HCPs found. So you can see there is a steady increase in the protein yield as more data is accumulated and processed by the software. So in this case, doubling the protein load on the column or doubling the repeats had a less significant impact than increasing the gradient runtime, which was approximately 15% longer. So using the appropriate LC method for your cell type will affect the separation of all proteins in your mix, and better separation usually equals better data. But the question is always going to be to what degree. So it's important to keep an eye on this and find a good balance between better data and then the time constraints that are associated with generating it. You can see as well that as well as producing higher HCP yields at this 1% global FDR, a longer gradient illustrated in the histogram will also generate significantly more peptides and therefore most likely confidence in the HCPs observed. So in this next example, we wanted to show the importance of monitoring reproducibility. This is especially pertinent in cases where you might want to perform comparison of batches across months or up to a year, etc. And it's important your experimental setup is practical and consistent. We've demonstrated this here with one of our standards, which is a BSA peptide, and that was done over the course of several projects where we were batch testing the HCP levels. We were looking actually at different sample types. So here we've got an XIC of the parent ion for the peptide and showing on the top trace the isotope distribution of this doubly charged ion. And then at the bottom, we've got a zoom of a carbon-12 peak displaying the same overlays across the months or across the sessions. So intra-session variation from 2 different client samples that were analysed on the same session or in the same session is lowest, as you might expect. The retention times and the peak areas are consistent, and the fragmentation data, which isn't shown here, was also very reproducible. So really, this is just a point about keeping an eye on your standard spikes. It's a good way to check for reproducibility, and these parameters are especially important when you're doing, for example, batch-to-batch comparisons of the same sample types for HCP levels across the course of, a year or whatever the time period is. And then I've got a final example here where we've monitored 2 different digest conditions for an in-depth, it was a full assessment client study. So that involved optimization of the LC-MS method as well. So by presenting the data in a histogram like this, we wanted to see how the peptides per protein against the number of proteins looked in both cases, in both digest cases. So here we've got the peptide count from decreasing to increasing order, going from left to right. And the good news for us really was that in both cases we could see that the greater than 10 peptide count HCPs were by far the highest number of proteins identified for the lysate. And so, this is what we wanted for a library in both experiments. It happened to be the case. As you might expect, when you put a higher load on the column, which is in purple, the HCP number does go up. And yeah, we wanted to check to what degree. So as a last point, we've written a manual review, and I just wanted to demonstrate how we do it to pick up for any errors or gaps using automated software detection. So at BPS, we start with an automated processing cleanup of the swath session export, and then we filter by CV, CV percentage, reproducibility, and scoring. And then we use a simple Excel table, which you can see on the right, where we've inserted the data into columns according to the HCP description details, peptide details, the accession numbers, the estimated amounts, although you can't see that there. And then we'll have 1 or 2 reviewers that are assigned to look at the raw data matches and make an assessment really based on their experience about the quality of the data and whether we think it's true. So sometimes we focus on specific elements of these data sets, for example, targeting just the high intensity hits, to weed out any of the false IDs that we think might be there. And even then, really, we need to say that no method is going to be perfect, so it's important to recognise that false positive identifications are possible, and also that identification lists might miss candidates. And so really, that's the reason why it's important to run ELISAs also, or other orthogonal techniques where possible. And in doing so, you increase your coverage and avoid any big HCP oversights. To summarise, the regulators want to see evidence that can corroborate HCP ELISA results. LC-MS provides a great complementary alternative that's accurate and sensitive with high reproducibility, and that can detect HCPs which are not limited to those that only produce immunogenic responses. So we've highlighted 3 areas for consideration when thinking about your HCP-MS analysis strategy. Spectral library types that determine the depth of analysis, sample selection, which is dependent on the level of screening that might be required, and then finally consideration for data expectations during the review process and optimization where needed. So the strategies and rationale for implementing HCP-MS will differ depending on the product, the process, and other project-specific variables. And we hope that by highlighting these different considerations, this talk has helped you to think about either adding MS to your HCP detection strategy or refining it further. Thank you for listening, and please see our details below for any further questions or feedback after this talk. Thanks.
Thank you, Dr. Morris, for your informative presentation. We will now conduct our second audience poll question. As a reminder, the question is displayed in the presentation window. Please click on the answer you would like to submit. If you are looking to use a CRO to support your HCP-MS analysis, are you most interested in A, quick overview of 1 or 2 samples to confirm ELISA data, B, full assessment to understand your process clearance, Or C, accurate assessment with method transfer to GMP compliant quantitation and batch release. Again, the poll question is, if you are looking to use a CRO to support your HCP-MS analysis, are you most interested in A, quick overview of 1 or 2 samples to confirm ELISA data, B, full assessment to understand your process clearance, or C, accurate assessment with method transfer to GMP compliant quantitation and batch release? We'll give you a few more seconds to make your selection in the poll. And it looks like our audience is most interested in B, full assessment to understand your process clearance. Again, thank you very much for your responses. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, up until now, the industry gold standard technique for HCP analysis is immunoassay, a high throughput and sensitive ELISA. You are advocating the use of mass spectrometry. Can you briefly summarize the pros and cons of each approach?
Sure. So if we start with the ELISAs, I think most of us know they're high throughput, they're highly sensitive, and they can be off the shelf, so with straightforward testing. A generic or platform ELISA will detect the majority of the HCP species, but it won't detect proteins that are specific to a manufacturing process, which is why people also develop process-specific kits. But these can be costly, so that's a downside, and they can take time. And of course, there's also still the drawback of missing any low reactive or poorly immunogenic HCPs. MS, on the other hand, can detect a wider population of individual HCPs within a single run. It's also highly sensitive. It identifies individual HCPs, sometimes down to sub 10 ppm or single digit ppm levels, depending on the MS methods that you're using and the instruments you're using. And it's also very flexible once you've actually developed the method. So the cons, I would say, for the technique probably include the fact that it requires skilled operators with good experience to perform the analyses and also to interpret the data. And for label-free quantitation methods, the HCP detection is dependent on the ionization capability of the protein fragments, and that was something that I mentioned earlier as well, which is a downside, but it's why people also they switch and use the more accurate labeling methods for MS.
Thank you. Our next question, is it possible to quantify all the HCPs in a biological product using MS?
All right, so the short answer there is no. We're not going to find necessarily all the proteins, which is why it's recommended as an orthogonal technique. But I would say, just as I've discussed with ELISA, the same is true for that kind of methodology. So MS does have incredible dynamic range. It can detect low levels of HCPs amongst the high drug product, but you can still have suppression of peptides of interest, or as I said, ionization inefficiencies, or perhaps you've got incomplete digestion or other interferences, which is why we always recommend using different methods to prevent any HCP oversights.
Thank you. Our next question is, are these MS methods acceptable to regulatory authorities?
Right, so in our opinion, the MS should be used as an orthogonal method, so it's an orthogonal approach. It does have the sensitivity and depth to identify differences with the HCP profile, and we would say really that it's probably best to use alongside an ELISA, for example, and to provide the broadest degree of coverage. So really, the advice I would say from the regulators should be that ultimately it's best to seek them to get proper approval and find out if this is something that is suitable for your process and for your product. It might change with time, but for the moment, it should be considered as an orthogonal technique.
Thank you. Next question. Are there any particular values for HCPs allowed during the production of biotherapeutics in different hosts and the accepted methods to quantify them?
There isn't a single universal test for HCPs. It will be specific, again, to your product. ELISA does remain the gold standard, and we would encourage the use of mass spec to cover any of the gaps and corroborate the data. In terms of the HCP numbers, again, it's process and product specific, but there's no absolute control limits. There are some factors to take into consideration. So I would say these include the maximum dosage of your product, the frequency of dosage, the route of administration, for example. Maybe if the therapeutic is topical versus an injectable. So the FDA more recently indicated in a report that the likely range of HCPs should be between 1 to 100 ppm, and some biotech companies are using that as a guideline.
Thank you. Next question. For the spectral library generation, do you perform sample pre-fractionation to improve your IDs?
Not usually, but if we believed that the LC separation was not good enough, then we would switch over to DLC methods for the library. Yeah.
Thank you.
So it's dependent on the sample. Yeah.
All right. Thank you. Next question. Are you running DDA runs prior to swath to generate a spectral library to match to?
Yes. So we're running the IDA because we're using a SciEx system, and that's to generate the targeted MS-MS data where we have a known precursor ion that gives us the MS-MS fragmentation spectra. And then we will use that to build the library. It gives us some more confident hits, as I've described, and we'll run the swath data and then match that to the library that we've made from the IDA. Yes.
All right, thank you. Our next question, how many IDA runs do you find are necessary to produce a proper spectral library?
I think that's based on, to an extent, on the host cell type, maybe mammalian versus bacterial, for example. But we also, I would say, it's also dependent on the type of analysis that you're wanting to do. So if you're looking at doing a full assessment where you're using a lysate, then it would probably be more appropriate to up the number of repeats that you're wanting to generate, just so that you're increasing the coverage and the depth, versus say, just a quick overview check, and then it's less critical.
All right, thank you. Next question. Are you using nanoflow or microflow LC-MS in these methods?
We use microflow most of the time. Nanoflow will increase, it'll be better for separation and for looking at these low-lying HCP numbers. So again, depending on your needs, really, you can tweak these methods and the LC conditions, etc., to improve your library. It is a balance, as I said, and it's just trying to work out what is appropriate according to what your kind of strategy is, I suppose.
Thank you. It looks like we have time for one more question. You showed a graph for the amount in parts per million, ng, mg, of HCP in different steps of purification. Can you share how was the absolute quantification done? Thank you.
Okay, I'd probably need to see which graph we were talking about there. Do we have a slide number?
I don't, and I'm not sure that we're able to.
Are we talking about the full assessment when we were showing clearance? So if so, then that wasn't an absolute quantitation. That was using swath with relative quant. And that's just sort of like the process from start to finish, where I was talking about it, I think, in the one of the early slides, maybe slide 6, where you start with your, in this particular case, we started with ELISA to make our reference library. And then we ran IDAs on our samples from upstream to downstream, and then we ran SWOS as well. And so the clearance that you're looking at in those bar charts is just an example from a client study where, as we went downstream, or as we moved downstream through the analysis process, we saw fewer and fewer HCPs. So that's what we've shown there. We've shown different sample types on the X, and on the Y we've got the amounts that have been quantified based on the SWOS data that was analyzed and reviewed.
Well, thank you, Dr. Morris, for your time today and your important research. We would also like to thank LabRoots and our sponsor, SciEx, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions and participation in the polls. Question we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.